Hi guys, today I'll be talking about DNA replication, a semi-conservative story of love. In scene 1, the DNA double helix is composed of two antiparallel strands, each strand made of numerous nucleotides. Each nucleotide is created by one 5-carbon sugar, one phosphate group, and one nitrogenous base. All of the nucleotide sugar and phosphate groups are bonded together by a phosphodiester bond, which occurs between the phosphate groups and the 5-carbon sugars of the nucleotides, making up what is known as the phosphate sugar backbone. There are 4 different types of nitrogenous bases, which you may know them as A, T, C, and G. A, known as adenine, is complementary with T, or thymine. On the other hand, C, known as cytosine, is complementary with G, known as guanine. Each pair is bonded together by a weak hydrogen bond, which is weak in order for replication to occur, which I will explain later. In scene 2, during the start of the replication of DNA, an enzyme called helicase, highlighted by the blue object right here, breaks open the DNA and makes a replication fork, splitting it into 2 individual strands. The strands are named differently. The leading strand, which goes from 5 to 3 in carbon numbering, is highlighted in blue. The lagging strand, which goes from 3 to 5 in carbon numbering, is highlighted in red. In scene 3, RNA primer anchors itself on the DNA strand and creates the RNA primer. The RNA primer allows nucleotides to be added on, or Okazaki fragments. On the leading strand, the RNA primer is situated far from the fork. And on the lagging strand, the RNA primer is situated near the fork. On one side note, there will be multiple RNA primers on the lagging strand, while there will only be one on the leading strand. In scene 4, scene 4, my bad. These Roman numerals are ridiculous. In scene 4, the DNA polymerase III, another enzyme, highlighted by the purple item, right here, attaches itself onto the DNA strand and starts adding nucleotides. Here, on the leading strand, the DNA polymerase III attaches onto the strand and starts grabbing DNTP that are floating around near the strands. The DNTP, which are just nucleotides with 2 extra phosphates, are grabbed on DNA polymerase III. The 2 phosphates detach from the DNTP, turning the DNTP into just an ordinary nucleotide. These nucleotides are added onto the leading strand by polymerase III. The complementary bases are added on as polymerase III moves towards the replication fork. However, on the lagging strand, nothing as of now has occurred. On the leading strand, the DNA polymerase III keeps on adding complementary nucleotides onto the strand. However, on the lagging strand, its process of replication has just begun. Another DNA polymerase III attaches itself onto the lagging strand and starts pulling in these complementary fragments called Okazaki fragments. This process, however, goes away from the replication fork. Due to this, more DNA polymerase III and RNA primers are needed. When all of this is over, the DNA polymerase I removes the RNA primer on both the lagging strand and the leading strand, completing the replication. Another enzyme, called the DNA ligase, attaches itself onto the lagging strand and glues the Okazaki fragments onto the lagging strand. Throughout the entire replication, the original strand is conserved, which makes the entire process semi-conservative, making the DNA replication semi-conservative. And that's it all for me today. I hope I don't bore you guys to death. Well, 4 minutes. Not bad.